How does stable isotope analysis determine trophic levels? Have you ever wondered how scientists figure out what ancient people ate? One fascinating method is stable isotope analysis, which helps determine trophic levels in food chains. So, how does it work? Stable isotope analysis primarily measures the ratios of stable nitrogen isotopes, specifically nitrogen-15 and nitrogen-14. This measurement is expressed as delta nitrogen-15. As you move up the food chain, the delta nitrogen-15 values increase. This happens because organisms tend to excrete the lighter nitrogen-14 isotope, leading to a higher concentration of nitrogen-15 in the tissues of consumers. On average, this enrichment is about 3:2 parts per 1,000 for each trophic level. This predictable pattern allows researchers to estimate where an organism sits in the food chain. In anthropology, this technique is applied to both human and animal remains. By analyzing the delta nitrogen-15 values found in bone collagen or other tissues, anthropologists can determine if individuals primarily consumed plants or animal proteins. This information is especially useful in bioarchaeology, where it helps reveal ancient diets and social differences in food consumption. But nitrogen isotopes are not the only players in this analysis. Stable carbon isotopes also come into play. The delta carbon-13 values provide additional context about dietary sources. They help distinguish between different types of plants, such as those using the C3 or C4 photosynthetic pathways, and whether the food came from marine or terrestrial sources. Together, these nitrogen and carbon isotopes create a clearer picture of dietary habits and trophic relationships. To accurately determine trophic levels, researchers first establish an isotopic baseline. This baseline usually consists of the delta nitrogen-15 and delta carbon-13 values of primary producers or consumers in the environment. This step is essential because natural variations in isotope ratios can occur over time and space. Without this reference, interpreting consumer values could lead to incorrect conclusions. In practice, stable isotope analysis in anthropology involves sampling human skeletal remains, animal bones, or other organic materials. The samples are then analyzed using isotope ratio mass spectrometry, which measures the isotope ratios with precision. The resulting data informs models that estimate trophic positions, dietary sources, and ecological interactions. By using stable isotope analysis, anthropologists can reconstruct ancient diets and understand the relationships within past human communities. In summary, stable isotope analysis determines trophic levels by measuring nitrogen isotope enrichment across food chains, supported by carbon isotope data and baseline references. This method is a powerful tool for understanding ancient diets and ecological relationships.